to introduce you the next speaker. His name is Dr. Sundar. He is the Institute Chair, Professor and Head in the Department of Biochemical Engineering and Biotechnology at the Indian Institute of Technology, Delhi, here in India. His areas of interest is very, very diverse. It is in the area of bioinformatics and computational genomics, and design and analyze genomic engineering tools, and develop AI ML based solutions, and that is involved in cancer research, and a whole list of them I won't have the time to go through. So with all that, a great pleasure to introduce the next speaker, Dr. Sundar.
Thank you very much for the kind introduction. Good morning, and first of all, I would like to thank the organizers for having given us an opportunity to present our research under the banner of Picnic. And the topic that I've chosen today is drug membrane interaction studies and how it can lead to new considerations for targeting cancer cells with selectivity. And we all know that development of drugs that can be highly selective for cancer cells is very important, which are safe for the normal cells. And one of the key parameters, one of the key metrics that one can look into is by evaluating the ability of the drug to permeate through a membrane. So selective targeting in cancer therapy is important because of the reasons what you can see. Many of these cancer chemotherapy is often associated with severe side effects, and this could be potentially because of the lack of selectivity. And studying the interaction of anti-cancer compounds with the specific targets, which are very unique to cancer cells, provides the leads for us to start with. And if there is a way in which you could study the interactions, the pharmacokinetic properties of these compounds in the tumor microenvironment, and that can provide insights for enhancing cancer cell selectivity. And we would like to project here some of our data from how we could get insights into the permeation of drugs which could be potentially useful for chemotherapeutics. So when you look into the selective targeting of cancer cells, right, one way of looking at this is when you look into the pharmacodynamics part of it, where we look into molecular targeting, which would be straight-jacketed work where you look into the target proteins that are unique to cancer cells. We also look at target proteins that are overexpressed in cancer cells, and here heterogeneity is a challenge. So the drugs that can target multiple targets have been reported to be quite promising. But I would like to draw your attention to the pharmacokinetic part of selective targeting of cancer cells, where we are looking into, I think you're able to see it clearly, let me see the slides here. So targeting the small structural or physiological differences existing between cancer cells and normal cells. Is there a way we could learn from that, which could be important for targeted delivery? And there are also reports on how a cancer cell membrane looks like and how that changes the dimension in which a potential drug could selectively permeate through a cancer cell. So what I'm going to present maybe in the next 5-10 minutes is the drug membrane interactions that we're studying and how we could identify some kind of a selectivity in the chosen drugs. So I'm not going into the detail of this particular slide. So there are quite a lot of interactions that actually can happen when a drug is actually in the membrane, and right, I think these are textbook information. But there are a lot of things that one can learn from how a drug interacts as it goes through the membrane, and important information related to absorption, distribution, metabolism, and so on. All these information can be easily derived today by performing computational experiments to derive some insights. And this is the barrier which actually needs to be passed by any drug molecule. And one can easily achieve many things, which is very, very important to understand the problem. So when you look into the membrane, and I think I'm not again going into the details, one of the important things that we need to understand is the asymmetric distribution of phospholipids. So the lipid bilayer which is there in the membrane and the composition of it. So there are quite a few that have been reported. Right, we have this phosphatidylcholine, then sphingomyelin and so on. So there are multiple such composition which is reported for membranes, and I think there are also varied distribution of these. And when you look into a normal cell and a cancer cell, there are quite variations that have been reported. So one of the things that we could do to understand how a drug permeates through a membrane is to understand the free energy profile. So if a drug moves through the membrane, the kind of interaction that happens, so on a scale, you can actually look at the interaction of a drug, let's say from outside the membrane, through the membrane and inside the cell. So there are ways in which these can be measured, a drug moving through the membrane, and we can calculate what is called as a potential mean force. So that basically tells us if you want to measure the permeation as a function of a distance or as a function of the different composition of the lipids which is going to be making these membranes. So today with modern computational techniques, one can do what is called as an umbrella sampling. So there are ways in which you can place a drug on, so you can build a membrane with whatever composition you want based on a particular composition. So here in this study, what we are reporting is a membrane which is largely composing of phosphatidylcholine as one of its major components, and that has been reported in several cancer cells. So you can do what is called as steered molecular dynamics. So without going into the details, so to put it in simple terms, so if you are going to have an extracellular site of a membrane, you can build a membrane with whatever composition you have. You can have an outer leaf, you can have an inner leaf, and then you can look at the drug passing through the membrane, and you can see what is actually happening to its trajectory from outside to inside. So you can actually have a force, you can actually push it from one side and pull it from the other side, and you can look at the trajectory. And this is what we look at, the calculation of potential mean force. And there are some standard measures where you could calculate the permeability coefficient and so on. And this actually, as I said earlier, comes from the kind of asymmetry that is lost in cancer cells. So when you look into normal cells, the distribution of the phospholipids on the membrane is pretty asymmetric. But this becomes quite symmetric in cancer cells, that is what is reported. And this is one of the data that we have used for preparing or building a membrane. And that is what I'm actually trying to summarize here. So there are ways in which, I think, this is a standard pipeline where you look at identification, hit identification, lead optimization. So this is where this kind of a study can intervene and throw some insights into whether the drug or the potential molecules that you have identified can really permeate through. Because there are a lot of papers which talks about how they interact, the potential drug target interaction and so on. But many such things fail. An evaluation of this parameter, whether it can pass through a normal cell or it can be very selective for a cancer cell. So this kind of a study which can provide insights into preparing cells with different compositions of phospholipids can be very, very helpful. So what we have done in this study that I'm reporting is, so we have made a composition of a lipid membrane for a particular cell membrane that looks like a cancer cell, which can potentially modulate the drug activity. And we have looked at ways, so this is one of the very first such reports where we have tried to build a membrane with different compositions. I'm not going into the details of the different membrane compositions, so I'm only presenting the data from one of the phospholipids, which is phosphatidylcholine, and the details are here. So the phosphatidylcholine is usually present in the inner leaflet of the membrane, so you can dissect the membrane into different sections, and you can create composition of these membranes based on what could potentially look like a cancer cell membrane. And that is how we did in terms of the number of molecules that were placed on the inner leaflet and on the outer leaflet and so on. And then we exposed this lipid bilayer membrane to a particular drug. We pushed it from outside and looked at its trajectory through the membrane and see whether it is able to enter into the cell. So, right? So we built this. So this is just a summary of what we have built. So we have built a normal and a cancer cell membrane based only on phosphatidylserine exposure. So there can be many different ways to make this. This is one of the pilot studies that we have started with, and we looked at, right, I think, the composition, and we also throw in some of the other key ingredients for a membrane. But the major thing that we are looking at is whether it can pass through this particular membrane. So as Dr. Vadva mentioned earlier, we have been studying on some of the natural compounds. So the ones that were chosen for this study included the ones from ashwagandha, which is vitaferin A and methanol. And we also looked at two other compounds, which is what is called as the CAPE, the caffeic acid phenethyl and the atropinin C. And I think the slide is not clear here, but basically we had some data in terms of some selectivity, which was actually obtained from experiments, where some of the natural compounds were highly selective for cancer cells and some of them were actually safe for the normal cells. So with this data, what we started was we built the membrane, and I think what I'm showing you here is the differential permeation, which has been calculated by the measurement of what is called as the potential mean force. So you're seeing these four panels: vitaferin A, methanol, CAPE, and atropinin C. And one of the blue color, light blue, the cyan is the normal cells, the trajectory of the drug through the membrane, and what you see on the red is the cancer cell, the membrane through which the thing has passed. And the first inference, I'd like to draw your attention to this peak here, which actually is very clearly showing the hydrophobic code of the membrane. So you can see, right? I think this is the x-axis shows you the length of the membrane, and what you can see here is the hydrophobic code of the membrane, which is seem to be creating a major hindrance. So this is for vitaferin A, methanol, and similarly the same is the case for CAPE. So there seems to be some kind of a hindrance there. And then when you look into what's actually happening to the atropinin C, it's kind of the head group, the lipid head group seems to be the major hindrance. So the principal hindrance for different molecules are different. So that means this will also decide the way the drug is going to move through the membrane. And similarly, we went on to measure some of the key parameters that are the free energy change and so on, which are generally used to measure how a drug is able to move into the membrane or it is not able to move into the membrane. And I think when we go into the other way, right, I think one actually we look into the diffusivity, which is the direct measurement of permeability. We also looked into the resistivity, in terms of how a particular membrane with a certain composition is able to resist the movement of the drug through the membrane. And that was also quite corroborating with what we got in terms of the permeation. So the summary is, if you have a membrane which looks like a cancer cell membrane, in this case it is largely made with one type of lipid, which is phosphatidylserine. It caused some change, so it's a very preliminary study. It changed the energy landscape, whether it is right in the middle or through the end of the membrane towards the inside. So the models that we have developed and we compared from the normal and the cancer cells, that clearly showed that there seems to be some kind of a selectivity of a certain type of natural compounds. In this case, it happens to be vitaferin A, methanol, and CAPE, where there was a differential permeability in terms of the normal and cancer cell. And this selectivity can be explained with the difference in the free energy landscapes and the water interface. So because solvent is also going to be one of the key players, so you could measure this as a function of distance or the different compositions of the membrane or even the environment where it is going to be. So this, though may not be a very complete study, but it really brings in some kind of an insight. So we started off with some experimental data that we had in terms of selectivity, and we were able to add more value in terms of how there seems to be a difference in terms of selectivity towards the cancer cells, which is very, very helpful. So to conclude, I would like to say that I think one can study different such membranes, build different such membranes to understand how drugs are able to move, and this difference can be very, very helpful in terms of optimizing the drug, right? In terms of, because we also, I'm not going into the details because the different compositions, each drug is going to have a particular structure. The different parts of the drug molecule interacts differently to the different composition of the membrane. So all such details can be easily be obtained, which can straight away tell you about the selectivity. So one can be even planning their experiments before getting into the nitty-gritties of whether it is going to work or not. So in terms of future directions, where we are heading towards this, we are trying to now build. So we have actually have many such models where we are trying to make with different lipids which have been reported in literature. So we are looking at, and I think one thing which we could see is there is not much literature on the composition of such cancer cell membranes. That would be very, very helpful. So these are being built in the light of these preliminary studies. So this has been recently published in journal Membranes, and different class of drug-like compounds can be studied, so that these can be used as a very, very important consideration before the drug design and further processes take place. Thank you very much, and I'll be happy to take any questions.
Questions? Yes.
Lipid membranes. And in cancer cells, you know that efflux proteins are very highly expressed in the drug. It's not a normal mechanism of movement of drugs. So how do you plan to address
So actually,
these?
So I think this, as I said, was only concentrating on lipid composition. There are also other reports that we have done where we have looked into all possible potential interacting factors. So that could be easily added to the membrane. So it could be done. So we can pretty much mimic a real membrane looks like. So the objective of this study was only looking into the lipid composition. That broadly tells you whether it's going to move or not. But then I think other factors will come in terms of its potential passage through the Yes. Because if you see, some of these things could not even move through a normal cell membrane. So that actually is a very good thing to start. It's a very valid point.
Hari has a question. Hari? Yeah.
It's a wonderful presentation. What a nice concept to look at differences in the membrane lipids, especially phosphatic serine, which is in at least 7.
Please speak for the microphone.
Which is at least overexpressed in 7 different tumor types you show. And I think if you can add, as was just presented, other proteins and glycoproteins which form the complex on the membrane surfaces, that will make this molecule. But beyond that, if you look at solid tumors, there is a high pressure intratumoral pressures, which is one of the limiting factors for our drugs to go there. So if we can model that and how we can use that information to have drugs more but wonderful model. Enjoy it.
Thank you very much for your Yes.
Can I ask?
Yes, please.
It was a wonderful talk, sir. My question is that different drugs
at different concentration might be acting differently over the cell membrane. So starting off with such an experiment, how do you actually decide that this is the particular threshold, or have you considered this thing a different concentration and this whole change that?
So I think, so in fact we have published a paper last year where we have, it's a valid question, so this has been one of the things that has been in our mind. So we have always been debating with our experimental collaborators about concentration. So I think concentration makes a difference in experiments. So how do you do that here? So we have looked into it. So when we have thrown in different populations of these drug molecules and we can track it. So eventually what we have seen is it is taking a similar path in terms of path, right? We have even looked into keeping them at different parts of the membrane and then trying to pull down. That has also not made a difference in terms of tracking. So the objective, as I said, here is only to see whether it is even able to move, right? So I think that is a very valid point, and it is there in our mind for quite a long time.
Thank you, sir.
One more question.
Thank you for the presentation. Cholesterol is known to increase membrane fluidity, and cholesterol is there in normal cells also. So how would you think cholesterol would make your results a little different?
No, we have added, so I have not gone into the details. Actually it is, so we have pretty much tried to add as much, so in terms of number of cholesterols, we have added all of this. So, so pretty much it is there in this time. So only thing is we have kind of played with how many molecules will be placed where, whether the outer leaflet and the leaflet. So that is something we have done quite a few background studies.
Thank you, Dr. Sundar, for such a Sundar talk. We all enjoyed it.